Another way to slow down release of a drug is to use implants. So, there are mini pumps which allow us to continuously administer the drug. So, for example, continuous osmotically driven mini pumps for insulin have been developed, which are implanted under the skin. So, what these pumps do is that they monitor the level of insulin in the blood and release the hormone as required to keep the levels constant. Right. So, one of the major problems with diabetes is the high level of fluctuation in blood sugar and insulin. And so, this large fluctuations that are going to happen can be avoided. And when insulin is administered through regular injections, the levels goes up substantially very soon, and this is also a huge problem. Gliadel is a wafer that has been implanted into the brain to administer anticancer drugs directly to brain tumours, and these can be used to avoid the blood-brain barrier. Okay. There are also polymer-coated or drug-releasing stents that have been used to keep blood vessels open. So, for example, when there is a procedure of angioplasty, the clot in the blood vessel has been cleared, and then after that what is sometimes done is to put a stent which has a drug coated on it. So, this will slowly release an anticoagulant, for example. Investigations are presently underway to use of implantable microchips which can detect chemical signals in the body and release drugs in response to these signals. So, these are on demand drug release type of situations. Now, let us discuss the topic of drug dosing. So, this is the amount of drug that needs to be given to a patient. Of course, we have looked at number of pharmacokinetic variables, and because of this large number, it is sometimes difficult to estimate the correct dose regimen for a drug. Now, how much of a drug to give, how often does one give it, all these are major issues. So, ideally, the blood levels of any drug should be constant and controlled, but this would require a continuous intravenous drip, which is clearly impractical for most drugs. So, drugs which we are commonly encountered or we are commonly used to are usually taken at regular intervals orally, and these are designed to keep the blood levels of the drug within a maximum and a minimum level. So, it cannot go too high, then what happens is that it can become toxic, and it cannot be too low either, which will make it ineffective. So, one of the parameters that we can measure is the concentration of free drug in the blood, right, that is not bound to the plasma protein. So, this gives us a good indication of the availability of the drug at its target site. Of course, this does not mean that the blood concentration level is the same as the concentration level at its target site, because we know that the target site invariably not always in the blood, and there can be variations. But any variations in the blood concentration will perhaps result in similar fluctuations at the target site. So, therefore, blood concentration levels are used reliably to determine what are known as therapeutic dose and safe dose for a drug. Now, let us look at an example where we are providing a drug at a particular dose. So, here is the plasma drug concentration, and here is the dose times. This gives you the number of doses. So, the first dose is given, and so you would expect the drug concentration to go up, right, and then due to metabolism and excretion it comes down. So, here is the second dose that is given, it again goes up, and then it starts to come down, and then here is the third dose that is given and again goes up, and then it starts to come down, and then here is the fourth dose given, again it goes up, and so on and so forth, right. So, what happens is that as the dose increases, you would have this type of a behavior, right. So, at this plasma drug concentration level, we can determine two important parameters. Let us say this is the dose at which the compound is effective, that is, this is the therapeutic level. So, you would want plasma drug concentration to be higher than the therapeutic level, but let us say this is the toxic level. That means, at this concentration or above this concentration, the drug is toxic. So, therefore, we would want to avoid this region completely and dose it such that you are able to avoid going to the toxic level. So, this window is called the therapeutic window. So, this regimen, which we are going to call as regimen A, here what happens is that it reaches the therapeutic level, but continues to rise to a steady state where it is toxic. Let us consider giving this drug at a lower dose. So, here what we would do is to give essentially half the amount. So, when you start out at a lower range, goes up and then comes down, goes up, second dose is given, comes down, third dose is given, comes down, fourth dose is given, comes down, fifth dose is given, comes down, and so on, right. So, here if you call this as regimen B, in regimen B it takes longer for the compound or the drug to reach therapeutic level, but it continues to be efficacious because it does not go into the toxic zone. So, by changing the regimen, one can figure out what is the right dosage to administer the drug at. So, dose B involves half the amount of drug provided with the same frequency, but the time taken to reach the therapeutic level is of course longer, but the steady state levels of the drug remains between the therapeutic and the toxic levels. So, dose regimens involving regular administration of a drug work well in most cases, except especially if the dose is less than 200 milligram and dose is taken once or twice daily. But certain situations such time doses are not possible. For example, insulin secretion by pancreas is nearly continuous, and so giving a dose is unnatural and will have to be done with care. Of course, there are other problems which are associated with age. So, the older the patient is, the more complicated the metabolism might be. There are gender issues, and we have looked at previously there are also race issues. And the diet, the food that is taken along with the medicine will have a profound impact. Of course, the environment and altitude can also have important influence on the way in which the drug is going to act. There is a class of population which are obese, and these present a very peculiar problem, because it is very difficult to estimate how much of the drug will be stored in the fat tissue. Okay? And because there is some of it stored in the fat tissue, and it is difficult to also estimate how much is free in the blood supply. So, the precise time when drugs are taken may be important, because the metabolic reaction rates can vary throughout the day. So, the next important thing about the journey of a drug is the half-life of a drug. So, half-life is the time taken for the concentration of the drug to fall by half. Okay? So, you take a dose of let us say 200 mg, which contains some amount of active ingredient. How much of that how much time it takes for that active ingredient to reach 50% of the original concentration, right. So, this fall or removal or elimination can take place through excretion or metabolism, right. And as you can imagine, it would not be linear, right. So, you are not going to have a straight line that it is going to go down with time, and the metabolism is going to vary from person to person. Some people may be excreting it out better, some people will have higher level of metabolism. But the drug half life is defined in the manner in which we look at the original concentration that the drug was given at, and what is the time taken for it to fall by half. There are some drugs that can linger in the body for a long period of time. Okay? But for example, if a drug has a half life of about 1 hour, right, then there is 50% of it left after 1 hour. So, in a period of 2 hours, there is 25% of the original dose left, and in the period of 3 hours, 12.5% remains, and so on, right. So, it takes a full 7 hours for the drug to fall below 1% of the original dose. Okay. So, therefore, once a drug is administered, it can linger in the body for a pretty long time. There are some drugs which are analgesic, for example, the opioid analgesic fentanyl. They have very short half life of about 45 minutes. Okay. And whereas there are drugs like Valium which have a half life measured in days. Okay. So, when the half life is in days, recovery from the drug may take a week or more. So, having one dose of a drug can take you many days to recover from that particular dose. Now, let us define what is known as a steady state concentration. So, of course, drugs are metabolized and eliminated as soon as they are administered. So, it is necessary to provide regular doses of this drug. So, we already looked at this therapeutic level. So, in order to maintain this, you need to be able to maintain a steady state concentration. Therefore, it is important to know the half life of a drug in order to calculate the frequency of dosing, right. And for us to be able to ascertain how to reach that level of therapeutic level and maintain these levels. So, here is an example of a drug. What has been done here is that you take various time points and you administer a drug and it's a number of administrations that have occurred. And so what you do is you measure what is known as a maximum level after, let's say, 3 or 4 attempts, and then you measure what is known as a minimum level. So this is the maximum level and this is the minimum level, and at an interval of 4 hours you measure the amount of drug, right. So in certain cases you will find that the maximum level and the minimum level, there is a fluctuation, right. So once you administer the drug, the maximum level, let's say, goes to, this is on the y axis is microgram per ml, and this is time in hours. Okay. So in the drug goes up to the highest dose that it can go up to is 1 microgram per level, and the lowest level is 0.5 microgram per level. So if you plot the maximum level, it goes over here like this. Okay. And similarly, if you plot the minimum level, it goes like this, right. There is a fluctuation in the level in the period between each dose. Okay. The level is at a maximum after each dose and then falls to the minimum before the next dose is provided. Of course, it is important to ensure that the level does not drop below the therapeutic level, but does not rise beyond a particular point, so that side effects are induced. So in this case, it would be ideal if the therapeutic window is somewhere here. The time taken to reach steady state concentration is not dependent on the size of the dose, but the blood level achieved as the steady state is. Therefore, the levels of the drug present at steady state concentration depend on the size of each dose given as well as the frequency of dosing. Before a drug is actually released into the market, there are clinical trials that are conducted. So here blood samples are taken from patients at regular time intervals to determine the concentration of the drug in the blood. This helps us determine the proper dosing regime in order to get the ideal blood level. So you don't want the level to go up beyond the the toxic zone. At the same time, you don't want it to be too low that there is no therapeutic efficiency. So there's something called as the area under the plasma drug concentration, which is called as the AUC, which represents the total amount of drug that is available in the blood supply during the dosing regime. We'll look at this concept later in the course. There is a concept wherein people do not respond to the same dose of the drug, which is called as drug tolerance. And with certain drugs, this is observed, and it is found that the effect of the drug diminishes after repeated doses. So what needs to be done is that you need to increase the size of the dose to see the same result. Okay. So this effect is called as drug tolerance. It is commonly observed in certain types of drugs, for example, in painkillers and so on. And there are several mechanisms by which drug tolerance can occur. So for example, it can induce the synthesis of metabolic enzymes which result in increased metabolism of the drug. So once the metabolism of the drug increases, then what you would need is a higher dose of the drug for it to reach the therapeutic efficiency. Another possibility is that the target may adapt to the presence of a drug. So for example, occupancy of a target receptor by an antagonist may induce cellular effects which result in the synthesis of more receptors. Okay. So let us say you have a receptor on a surface on which you want to have an antagonist. Now instead of having 2 receptors, you may have 5 receptors that are produced, right. So this, what it does is that you need a larger dose of the drug to achieve the same effect, to antagonize. There is a concept called as physical dependence, which is associated with drug tolerance. So it is a state in which a patient becomes dependent on the drug in order to feel normal. Okay. So if you remove the drug, then there are lots of uncomfortable withdrawal symptoms. So you would have heard, for example, there are drug addicts who are extremely uneasy in the absence of the drug, right. And you would need to give the drug to the person for alleviating the symptoms. So these effects can in part be explained by drug tolerance. So for example, if the cells have synthesized more receptors to counteract the presence of an antagonist, now if you remove the antagonist, the cell has too many receptors. Okay. And so what happens is that this results in what is known as a kickback effect. That means there is a stronger effect now because the cells have become oversensitive to the normal neurotransmitter. Okay. And this is what produces the so called withdrawal symptoms. this will happen for some time until those excess receptors have been broken down by normal cellular mechanisms, and then it restores normalcy. But this process may take several days or weeks, and so the withdrawal symptoms, which are extremely uncomfortable and sometimes very difficult to overcome, may take several days or even weeks. The next concept that we need to understand is bioavailability. Okay? So, bioavailability refers to how quickly and how much of a particular drug reaches the blood supply. Right? So, once you have taken care of all the problems associated with absorption, distribution, metabolism and excretion, then bioavailability is the amount of drug that gets into it and how quickly into the blood. So, oral bioavailability is the fraction of the ingested dose that survives to reach the blood supply. Right? So, let us say you take 100 microgram and only 5 microgram reaches, then the oral bioavailability is 5 divided by 100, and it is the fraction of the amount of drug that reaches the blood supply. This is a very important property when it comes to designing new drugs, and of course, it should also be considered very strongly along with pharmacodynamics, that is how effectively the drug interacts with its target. Another important concept in this part of the course is formulation. So, this is the way in which the drug is actually administered. So, sometimes a drug is formulated in such a way that it can avoid some of the problems associated with oral administration. Okay. So, as you know, drugs are normally taken as tablets or capsules. Tablet is a compressed preparation that contains about maybe 5 to 10% of the drug and about 80% of other fillers, and there are some disintegrants, lubricants, glidants and binders and so on, and about 10% of compounds which ensure easy disintegration, disaggregation and dissolution of the tablet in the stomach or the intestine. This is a process which is defined as the pharmaceutical phase of the drug action. Of course, we will not be spending much time on this part of the course, and there is an entire industry which revolves around how to be able to deliver the drug or administer the drug in a very efficient manner. So, there are pills that can also be coated with sugar, varnish or wax that helps us disguise the taste, because some of the drugs are extremely bad tasting, and this helps us ensure that the drug is properly consumed. Many of us have taken drugs in a capsule. A capsule is nothing but a gelatinous envelope which encloses the active substance. Okay? And this capsule can be designed to remain intact for some hours after ingestion in order to delay absorption. Okay? You can also have a mixture of slow and fast release particles to produce rapid as well as sustained absorption in the same dose. There are a number of factors such as particle size, the form of crystal, that can significantly affect how well the drug dissolves. Okay? So, sometimes fast dissolution is not ideal, and you would like to have slow dissolution, and this will avoid high levels of the drug in the plasma, and it can also help us prolong the duration of action.